So, does it work? Yeah, seems to be. Yeah, after 1 week, it gets more used to the technical aspects. So, welcome back to the lecture. I will continue with a quick summary of what we did last time and finish the lecture because I did not fully finish. Maybe I switch on these lights here as well. Frente, okay, yeah, Frente is front. Okay, good. So what did we do last time? I mean, we want to explain essentially this interesting movie from this simulation here that describes the kinetics of an emulsion in a spatial heterogeneous system. The heterogeneity had been created by a quench of the equilibrium concentration, C out, it happens roughly now. And then, of course, the concentration was pushed down to the equilibrium concentration at each position, and that led in the end that the droplets here were eaten up by the droplets here because, yeah, they were simply bigger because the quench allowed them to grow bigger. This is called position-dependent Oswald ripening. In addition to this position-dependent growth, you also have drift, but also you have even shape modes. The shape modes is a bit too much for the lecture today, but at least I can tell you how to get there in case if you're interested. So this is what we did last time. So we wrote down, in the spirit of Lifshitz and Sliusov and Wagner, a ripening theory where we started with a single droplet analysis. So this is a single droplet, and we wanted to account for the asymmetry between the back and the front. Here I just called left and right because of the sketch. And there are two asymmetries. One is asymmetry is the far field. When you go far away from the droplet, you have obviously a different concentration field than here. That's the dashed line. Yeah? And overall, the leading order to greater heterogeneity is obviously a linear one. And the other heterogeneity sits here at the interfaces. So the front, the back sees two different equilibria, which comes with two different equilibrium concentrations. Remember, the EQ was the one that also encoded the effects of finite size. So always this multiplied by this Gibbs-Thompson relationship gives you exactly the actual finite size equilibrium concentration at the interface. Then we solved with a quasi-static approximation, essentially a Laplace equation, which just assumes that the interface grows still slowly enough such that at each moment in time the profile is stationary. It does not mean that there's no time dependence. That's why it's called quasi-stationary. It's not a stationary state. And this, of course, you know all from your electrodynamics lecture. Depending on the coordinates, here we have rotational symmetry. We are left with the radial part and the Legendre polynomial part. And we can solve this one. I do not write the solution again. Then we can calculate the flux at the interface right outside. Inside, we assume it's constant, simply because we assume droplets are far apart. Diffusion inside is fast enough to homogenize the profiles, but far away, there's diffusion, which is always heterogeneous. Yeah? And then you see, then you can use Fick's law, evaluate it at the interface, and calculate the interface velocity. We invoked here also two other assumptions. When the concentration is approximately constant inside, we have no flux inside, and the concentration inside typically is much bigger than the one outside, so we can neglect this one here. And then we obtained this beautiful expression, a bit lengthy, for the interfacial velocity. And sorry, when I wrote it down yesterday, I forgot this 1 over R factor, just in case you have recognized this. And there are essentially, one sees two contributions. So this is exactly the contribution we saw already from classical Oswald ripening, except that the far field concentration and the equilibrium concentration depends on position. Yeah, so it can be different at different positions. Yeah, okay, I just realized, okay, I apologize, just when I was writing it down. Maybe I write it here X now. To avoid inconsistency, because the movie in the column along the heterogeneity occurs here in the movie I called X over L, so it's X, and in the lecture sometimes I call it Z, but don't get confused now. It's just a clash of notation. Good. So this we have seen already, and then there's a new term here that scales with a cosine theta, and we argued a little bit yesterday already what this could be, and this likely gives rise to a drift. But to see that, I would like to show you a little bit how to do this more rigorously. So we have to somehow think about a shape parameterization. Yeah? And of course, in general, when you have non-spherical coordinates, non-spherical symmetry, this is a bit tricky. But if you have radial symmetry, so which means surface tension is strong enough that close to the droplet, we have still a radial symmetry, then I can define a shape function, which I here use with this kind of more curly shaped R, which depends on theta and time, yeah, and this vector here points proportional to the unit vector ER. Okay, I can decompose these shape functions into proper eigenfunctions, and in the eigenfunctions, I mean, when I think about the shape functions, we know already these are the Legendre polynomials. I can add here a coefficient. We talk about this coefficient in a second. I call it di. And the Legendre polynomials usually written with pi. And the argument here is cosine theta. Yeah? Good. Then we can link this nicely to the interfacial velocity, that if we are radial symmetric, this is simply the time derivative of this shape parameterization. And then we can just plug this in. Here we separated the geometric part with the time-dependent part, and we get a very nice definition here of the interfacial velocity, which is simply the time derivative of di. Yeah? This I will call now ui. Yeah? Good. So, how can we obtain these? We can also see them as moments, if you like. Yeah? And this is simply by a scalar product. And just to remind you quickly, when I have a general scalar product between two functions with Legendre polynomials, we integrate from 0 and pi. And because we had dx and x was cosine theta, we get from the Jacobi determinant sine theta gh. Okay, so this is how the scalar product is defined. Okay, then what are now these specific moments? We can write, for example, I equal to 0. Okay, I equal to 0 is d naught. Yeah, well, what is this? What do you think? What is d naught? So the zeros polynomial. So what is the Legendre polynomial? Is a constant 1, the first Legendre polynomial, and then it's cosine theta, right? So if I project this on 1, what do I get? I get this, right? What is it? What could it be? What type of object could it be that characterizes the shape? I mean, we had before, this we have learned as the change of the radius, remember that, right? But now it's just local. Same thing here. So R, I can identify with d naught, that's the radius. And I can define it with these shape functions simply as the scalar product where with the zeros order Legendre polynomial, and of course divide, because in general I get a scalar contribution from the projection. And then I can simply also write the time derivative of this. The time derivative of d naught is then the time derivative of R. And this gives you droplet growth. Yeah, that's how to calculate this. And this is a very systematic way, because now we can go to I equal to 1. Okay, what do we have? We have d1. What could d1 be? So So what you do is now you average the front and the back equally, and it's only non-zero if you do what? If the droplet drifts. Yeah? So that is x naught. Remember, x naught was here the drop center. Yeah? Again, I can rewrite this in terms of this scalar product by just taking this shape function R, projecting it on the first Legendre polynomial, and dividing by the corresponding scalar product. Oops, P1. And this gives me, again, a time evolution equation for my D1, which is exactly the time evolution equation of x naught. And that gives you droplet drift. So what is your expectation if we had looked at I equal 2? So the zeros mode was growth. The first mode was drift. What could be the second mode? Yeah, when you some people, yeah? Or you can just call it in, yeah, but yeah. Oh, yeah, okay. You can also say it, and then I can also say it to you.
Deforming?
Yeah, exactly, deformation. So it's the L equal 2 mode, you deform it in one direction. You make an ellipsoid out of a sphere, and then you can keep on going. Then you can get asymmetric modes, L equal 3, etc., etc. We don't have to write it. We can write it if you want, but we don't have to. Why do we not have to? We just look at that equation. When we do the projection, what are we left with? So we have one term that scales with, has no cosine theta. So it scales with Legendre polynomial P0. And one that scales with P1, that's cosine theta. So we have no contribution to the shape. And the reason when you redo the calculation is because we stopped accounting for linear changes in position of the equilibrium concentration and the far field concentration. If you had accounted for, in general, the next order, the second order, so in the end x squared or z squared type of changes, then you would also get deformation modes. Okay? But here in that case we don't get it. But of course we can identify now what this is. So this one here is then DTR, and it's a bit, and this one here, is this correct? Yep. This is DTX naught, and to be a bit more careful, I have to multiply then by D CN 0, yeah, because I don't want to have the cosine theta, that gets projected out, okay? So you see we have these two contributions, a growth term and a drift term, and that's how to do it in general, how to thoroughly identify their respective modes of the problem. Good. This is it. This is a single for the single droplet. We can now write an equation for many droplets, and that's exactly how this movie is created. Yeah? And then we are done with this lecture. So exactly that connects to what you have already seen in the lecture up to now. When droplets are far apart, okay, then it is as if the droplet sees the shared field. That's the far field of the other droplet that we call C infinity. Yeah, now infinity, the infinity index doesn't make any sense because there is no infinity anymore. Infinity means just, yeah, the field has relaxed to that field, and then the other droplet sees exactly this field somewhere here, and that's how they communicate. So this is exactly the leading order contribution when droplets are far apart. And of course, again, you can also make this more accurate and account for correlations when droplets get very close, but this is beyond the lecture today. So how are they coupled? Simply because there's particle conservation, and I can write down a transport equation, a PDE, now for this shared field. Now it's a PDE, so write partial C infinity. The field has to be able to diffuse. Remember, diffusion of the outer field we get by linearization. Yeah? We have just a 1D system. Yeah? So it's a classical 1D diffusion contribution. And then, of course, the droplets act as sink and sources, and this term we have already seen in the introductory lecture. So we have to sum over all the droplets, n. The number of droplets can change in time. Each droplet here in this simplified theory is considered to be spherical. This here is the volume change. Okay, I put already the 4 divided by 3 times pi in front of it. And of course I have to somehow tell, because here there's no, the I is set somehow the position of the droplet. I have to add here some sort of delta function that makes sure that the sinks and sources appear in space where the droplets are. Yeah, so that's, oh sorry, it bends a bit. So this is exactly the extended transport law to the classical Lifshitz-Luther theory, where there it was just an algebraic constraint. Now we have a transport equation. And then you can write down the same laws. I mean, I don't do it now. So we write down the same equation that I wrote down here. The only difference is we add subscripts I here, because we don't have a single droplet anymore instead of a not here. So it's the same equation. This gives you a set of PDEs you can solve. Diffusion accounts for an effective stochastic effect from already the transport. However, we can also make it truly stochastic. This was the question we had last time. You can also have here stochastic terms on the right-hand side. We can talk about this one in case you find this interesting. But this, of course, requires droplets to be small enough to be only composed of a reasonably low number of particles. If they're contained of 10 to the x particles, I mean, then these fluctuations are in general negligible. Good. Yeah, and now, so you could in principle sit down and implement this on the computer, because this is a classical diffusion equation with sink sources. These are kind of just extra. We have a list of particles, of droplets, they follow this equation, and you can reproduce movies of that kind. Of course, you can also visualize them as balls if you want, for illustration reasons. Good. Yeah, I think this is the minimum thing I wanted to explain to you. And so in case you have questions now about that, I'm ready to take questions.
Well, so when you take I equals 0, it's droplet growth. I equals 1, it's drift. I equals 2, it's deformation. And what is I equals 3?
Yeah, it's an asymmetric. I mean, in the end, you can think about, say you have a droplet and you deform it, with the, I should have not used I. So this I is not that I, okay? Just in case. So you just another index would be good. So, so if I equal 2, then you make an ellipsoid out of it. So if you have I equal 3, or I equal 4, maybe I equal 4 is easier. Then you make out of a sphere, you make something like this out of it, and this is just the asymmetric mode in between. It's just like, in the end, you can see them. I mean, the Legendre polynomials always have to obey this rotation symmetry. That's the only thing. But in general, when you decompose shape into the spherical harmonics, you have exactly those modes. So in the end, we only look at those modes that have still rotation symmetry. That's the only constraint.
Okay, thank you.
Welcome.
Hi. In the start, we assumed a linear concentration of particle R.
Yes.
How dependent is this behavior on that? And has it been tried with other functions?
Exactly. So you can go simply the review article, you find this extra material, active emulsions. There I looked at the second order. So instead of linear term, Z or X, sorry for the hiccup of the notation, I looked at Z and X squared. Yeah? And then you see deformation modes. Exactly. So in general, you could argue, okay, well, any concentration profile should have also nonlinearities beyond a linear one, yeah, and then they all exist. But of course, the deformation mode, there's something that fights against this deformation mode, and this is surface tension. So it's up to you then to think about, okay, how, I mean, it just wants to deform, okay, it can't, I mean, well, let me say it differently. A linear gradient, it can't deform, that's the point. But if you have a higher an harmonic contribution, then they can deform, but whether it deforms, it depends on how big the surface tension is. Yeah, because that fights against this. So you can even get rid of that by saying, oh, even a quadratic concentration profile, you just have spherical droplets because the surface tension is large enough. However, drift and growth, no way. It always happens. Yeah. Good. Any other questions? No? Looks good? Yeah, then let's do something different. Yeah, let's switch to the next lecture. And it's about chemically active droplets. I will show a few slides, and then in between do a few calculations. So the new thing we have to essentially calculate is the effect of chemical reactions. And this context of bipolar condensate is, of course, very important because the cell is not just at zeroth order a bag of molecules, it's a bag of reacting molecules with very fancy functions. Good. The way I would start with is a very simple-minded reaction. We can go far beyond that if you like. Yeah. But for the sake of the lecture, I always go with two-state transitions. So this is kind of the physics-minded simplest chemical reaction you can think of. And I call always one species sometimes an activated or deactivated molecule, and this becomes clearer later in the context when we talk about out-of-equilibrium driving. And what I will do, maybe I'll just go back one step. I will always consider that the states, according to thermodynamics, are not equally likely. So that in the end, for example, thermodynamics tells you that you hang around in this deactivated state. So think about a classical mass action law that you've learned in your lectures. If you calculate the concentration of A at thermal equilibrium compared to B, you will see it's mostly in the A state, okay? However, and this is the entire idea of these, what we call active droplets or active emulsions, you open the system and maintain the system out of equilibrium. And then for that, we often use fuel. Fuel is just a buzzword for using a high energetic molecule that gets effectively hydrolyzed and converted to a waste, and each chemical event gives an enthalpic energy that is far beyond kBt. Yeah? A good example of the cell is ATP. The hydrolysis of ATP to ADP gives roughly 10 kBt. So if you put this into a Boltzmann factor, thermodynamics is switched off for that event, for sure. Well, it's not relevant. And that's a bit the setting you should think of, and that's remember in my colloquium talk, we had a system like this. So if you maintain a system out of equilibrium, you always have to open it. There's no alternative way, no matter what your people tell you. So you have to open it and allow energy and or transfer of mass, essentially. So you need fluxes. And if we think about living organisms or living cells, that's exactly what we do. I mean, we are definitely not a closed system, right? We eat and we remove waste and continuously, and that maintains all the cells in your body out of equilibrium. Yeah, and that's exactly, and I switch maybe to the board. That's a bit the recipe I will use today, that there's an asymmetry where these activation processes occur. So when a system phase separates, essentially gives rise to coexistence of these two domains of different concentrations. Say this is B rich and A rich. And if you make it and design that there's an asymmetry, meaning, for example, inside dominantly B gets deactivated, yeah, while outside A gets, for example, B gets activated, yeah, then you see a range of very exciting effects. The movies, yeah, of these effects, it depends on you, whether you want to see the lecture first or the movies. Movies first? Okay, movies first. To see a reason why to listen, I completely understand. So then we do as if we fully understand what's going on, and I show you a few movies. So what can happen? Okay, this is now superficial in the sense that I don't explain why it happens. So you can get, okay, that's not yet a movie, you can get emulsions where droplets are all of equal size. So remember my introductory lecture, this is not possible if the system wants to go to thermal equilibrium because there's Ostwald ripening, big droplets eat up smaller ones, so you have this broad distribution, it's impossible. But you get in the end monodisperse emulsions, and they're completely stable, but there are fluxes in the system all the time that maintain this state away from equilibrium. Then you can have dividing droplets. So the droplets, they can grow, and this is just, sorry, this doesn't look like a droplet, just for representation reasons. This is a droplet, it's just cut that you can see into the inside, okay? And this droplet separates into two. So it does division. That's weird. So I told you, first of all, you bring two together, same volume, they fuse. That's a thermodynamic pathway, right? So you add up the two volumes with a new droplet, the total surface area has reduced, which means the total free energy of the surface, associated with the surface. So that's why droplets undergo fusion, but that's the opposite pathway. Yeah? And it did not revert the time here. Okay, that's already the explanation, sorry for that. Then, this you have seen in my talk, this is again a cut. They can form shells. Again, you form an inner interface. Thermodynamics tells you this is not possible, but in my colloquium talk you have seen this is indeed possible. And then I've shown you already this movie, for example, where you see even states that keep on bubbling around forever. Also this one should not be possible at thermal equilibrium. And indeed, it's because it's not should be, it is not possible. And for that, you need a continuous turnover of mass from one molecule to the other one, and thus also energy in the system. Good. Now I just, okay, this is, okay, no, this we can skip. A few more. Okay, here maybe I show you this movie as well. Okay, that's also a nice movie. Yeah. Okay, sorry, I forgot to add the division movies. But in case you're interested. With a little bit of luck, I will talk about this on Monday, but I'm not sure actually what we'll talk about on Monday. What can also happen is that you can have droplets that form, grow, shrink, and form, dissolve, and then form again. This means they oscillate. You see this? It comes again, and we do forever. And I think you have seen lots of, in pattern formation, lots of oscillatory phenomena, like a classic chemical oscillator, but in that case, it's kind of an oscillator of forming a droplet and dissolving. Yeah? And it keeps on going forever. Yeah? Also a phenomena that is not possible at equilibrium. Good. In my lecture, I would like to start explaining the fundamental prerequisite, yeah? And this is to understand that there is a length scale selected in the system that wants to fix the size or the diameter or the size of the droplets. Yeah? All the other phenomena are in some or the other way related based on this length scale selection. Yeah? So I cannot explain maybe all the different phenomena. The shells, I guess, I've explained a little bit in my colloquium talk, and today I would like to explain you why is it possible that you can get a monodisperse emulsion when you maintain chemical processes in phase separated systems out of equilibrium. Okay, and for this we have to touch base with a concept called detailed balance of the rates. Is this a concept I have to explain, or do you say like, no, we heard? Should I get into this in detail? Yeah. Okay. Good. Oh yeah, thanks. Oh, thanks so much, Ricardo. So maybe I clean the board because this was the old picture.
Chris, there is a question.
Hi.
There's a question. Yeah.
These patterns that show that the droplets forming like holes inside the bubbles, you can also have that on emulsions, or
I mean, of course, you can have many droplets where each droplet is doing this. You saw in the experiment, you saw this actually.
No, I mean, theoretically, you can capture this already, or
Yeah, but in the theory, we just keep things simple. So we just look at a single droplet and looked at it inside. If you had more computer power, we just make the system bigger, place 10 droplets, and each one is doing the same thing. That is so, because the interaction among the droplets is not the big thing. It's just it's a local instability that happens in each droplet. So in the end, but you're asking a tricky question, which we have not yet fully investigated. If you have multiple of these fluctuating morphologies, is there some sort of Oswald ripening still going on of the bigger droplets? We have not investigated it carefully. If I had to guess, I would say no. But to be fair, I mean, the numerical complexity of you making such movies is very high. So you would not say like immediately start with 5 drops. So we say start with 1, make it big enough such that this happens. Yeah, but good question. Yeah, please.
just a follow-up question.
Kill everything. Yeah.
This can only happen with bytes per system, right? Like a mixture.
A little bit louder, again.
It can only happen in a mixture, right? The bubbles, or I didn't understand.
What's the alternative? I mean, you need a mixture to phase separate, right?
Mm-hmm. Like, okay, yeah, that's true. Yeah, sorry.
Exactly. That's prerequisite. So of course there's other work you need in a way when someone writes only one field, like remember the first series I wrote, which has one field phi of x, it doesn't mean it's just one component. There was just another component, the other component was just 1 minus phi. So just be careful. We just removed, because of incompressibility, 1 degree of freedom. This was very helpful, but it's always a mixture. So phase separation requires okay, this statement is not true. Phase separation does not require a mixture necessarily, because you can think about a gas-liquid coexistence as well. But this is not what we investigate. Yeah.
You discuss always about emulsion. So what happens if you have a microemulsion? So meaning you have a lower surface tension. All this business is gone, or is still more complicated?
Yeah, I think that maybe also we should discuss about how this is exactly defined, because the microemulsion in some way is metastable in that sense. In a way, what this system is doing, it would do a macro phase separation, but it creates a micro phase separated state, but because of the chemical reactions. If the base state is a microemulsion because of also what you just mentioned about because of surface tension, I think we have not investigated that, honestly. It's an interesting question. Maybe let's talk about this after my lecture, yeah? Definitely good point, yeah?
One more question.
Sorry, I think I remember if you mentioned it, how you add the noise for the movies. It's like a Langevin equation or something?
Exactly. So, so of course, when you see these movies done with phase fields, or really the time evolution of concentration fields, there's a well-defined way how to do this. If it's a closed description, we know we have in general, we have some sort of multiplicative noise that we know how to add. The prefactor is fixed by the fluctuation dissipation theorem. That can be discussed. So if you maintain now out of equilibrium, so you hydrolyze energy by these chemical reactions, is this the dominant noise? Can be discussed. There are alternative ways how to derive it, from population dynamics, etc. This is what we're doing in the group. It's a big debate. What is the right noise? But say you know all the degrees of freedom. Say how to solve this problem is to write down a closed theory. In a sense, you write equations for the fuel and the waste. Then you know what to do. Then non-quantum thermodynamics tells you exactly what to do. And then you can integrate out these degrees of freedom of the fuel and the waste. And then you get an effective noise, which is in general multiplicative, so it depends on the state, and you can then solve these equations. But of course, multiplicative noise is per se a bit difficult, but yeah, this is what we do in the group. Exactly.
Thank you.
Yeah, but it's a very subtle thing. And of course, I wanted to talk first of all more about fluctuation, because this is what we're doing actually in the group at the very moment a lot. But it all relies on these deterministic concepts. So you need to know those to understand the stochastic aspects. So in case you're interested, I'm happy to give you more details. Good. Yeah, but I assume somehow a little bit that how to describe the non-equilibrium thermodynamics for open systems with chemical processes is not known to everyone. So let's start with this lecture 3. I call it, let's just call it active emulsions, or active droplets. And to get to the point, I would like to first discuss reactive mixtures, so mixtures with chemical reactions without fuel, okay? I think this you need because I use this fuel molecule to maintain the system out of equilibrium, okay? I use particularly fuel because something has to get, you have to degrade somehow the free energy of this molecule to use it for another process. So if you would say like, yeah, I just add enzymes, what does that mean? We need to be careful because an enzyme per se is not an active process, but many enzymes, many people think about in biology, also hydrolyze ATP, for example. So this is what the people usually mean. An enzyme itself does not maintain the system out of equilibrium, but in the presence of some fuel molecule, of course it does, and that's why I highlight this one here for the out of equilibrium nature. Good. Let's discuss homogeneous systems first. And we discussed this very simple reaction, this kind of, also when you think about proteins, for example, it can be a folding transition, a state of change. Yeah, I mean, physicists love it because it's the fundamental statistical physics model of a two-state system, like the Ising model. Yeah? I stay, for simplicity, in the canonical ensemble, which is in general with chemical reaction not very smart, but for the simple AB system we can make it work. Chemical reactions, please, are typically described in the Gibbs ensemble, and there's a good reason for that, because chemical reactions are not volume conserving. For the lecture, I assume a volume conserving reaction, so we're good. Which means if an A goes to a B, and the stoichiometric coefficient here is 1 each, this molecular volume is this molecular volume. Otherwise, I have a problem, okay? Good. Equilibrium corresponds at fixed temperature and volume to, so the total differential at fixed temperature and volume is exactly our exchange chemical potential times DNA. Sorry. And at equilibrium, this we discussed already, this is 0. But we didn't do this last time. When we phase separated, we minimized it with a constraint. Now I don't have a constraint. Why I do not have a constraint now? Because when I draw a free energy, let's say we again get this free energy, this led to phase separation. I plot here the volume fraction. Remember this simple Floer-Huggins model. You can now walk along the axis with the reaction because phi is not conserved. Yeah, because you can turn over A to B and vice versa. So if you start somewhere here, for example, you just walk. But where do you walk? Where do you go? Before we couldn't. The system had a conservation law. If I started here with my average, I had to phase separate into 2 phases. This was my constraint. Now I can walk around. Where do I go? Yeah, exactly. Yeah, exactly. You go to this global minimum here. Here's a local minimum. You get stuck there maybe for some time. Yeah. So this is my equilibrium state. Exactly. Yeah. That's a problem because that system, when I let it go, remember the Maxwell construction was something like this, right? Here it was a tangent, right? So it's outside that one, yeah? So the phase separated domain was this. This was the phase separated domain, yeah? Phase separated. So phase separation can occur here. But now we are outside this one, yeah? Which means when you add this chemical reaction to a system that could phase separate because it has this funny shaped free energy density, it would not do it. It would just go to the global minimum and it's gone. Yeah? So, actually, it was this very nice first work from David Zwicker. When was it? Some time ago. I add the date later when I find it in my notes. That the idea, okay, come on, let's maintain the chemical reaction out of equilibrium and rescue the droplet state. So that the droplet becomes truly active because you open the system, you add this fuel, remove the waste, and then allow it to hang around here, but you have to push the system in this direction. Okay, this was a cool idea. And this we do in a second, and it will actually lead then to this mono-disperse emulsion. Sorry, yeah, maybe here, of course, here, now I see it. The first reference, it was the original reference, 2015. Yeah. There's also a paper from Sharon Glotzer, actually, which is much older, which did the same thing, but in a different with a different purpose. But that was I should have added this here, I just realized. Good. So, equilibrium. So how do I use this constraint? Yeah? So anyone who discussed chemical reactions in your physical physics lecture would say, oh, now we have to introduce a concept in chemical reactions that's called the reaction extent. Yeah? So just for the professional people, what you would do here, you would and this has to do with the fact here's a continuum variable, here's a continuum variable, and the change of particle numbers in a chemical reaction is discrete. What does the differential of a number of 1 or 2 mean? So a bit like it's weird. So this is in physical physics, usually people don't think about this dn so much, but n is this integer valued, right? Okay, it goes from a huge number to another huge number plus 1. You think, oh, fine, there's a differential. But if I go from one molecule to another molecule, what does the DNA mean? Yeah? So anyone who wants to know this, I can tell you how to do this. Here we can stay a little bit, because that brings in the so-called stoichiometric coefficients here, the stoichiometric matrix. Here my stoichiometric matrix is a bit simple. In my case, it's just 1. Yeah? So indeed, you could say like, yeah, what is the DNA? If I ignore this integer thing, what can it be? It just gives me a 1 likely, and it does. So the equilibrium, the shortcut is here, okay, fine. To make this differential 0, this exchange gamma potential has to be 0. And this is actually be true. However, it's makes a bit of a jump. And if you want to see how this works, approach me after the lecture. Good. How to do kinetics with such a system? So dynamics, in that case, towards equilibrium. Just to distinguish this is called, we called it phase equilibrium, and this has also a different name. It's called chemical equilibrium. So if there's no other process happening, this corresponds to thermodynamic equilibrium. So for a homogeneous system, so what is the concentration? Remember, the concentration was always I over nu I, okay? I can write now for this simple system kinetic equations. It's homogeneous, so there are no fields up to now. It just depends on time. And obviously, when you think about this one, when I produce an A molecule, I had removed a B molecule. So you get here a minus, very naturally, a slave relationship. And then you can decompose this equation into what you can call and a backward pathway, okay? Yeah, so going from, this is really from A to B is the arrow to the right, or from B to A is the arrow in the other direction, okay? Now we can ask, what are those, right? So these are the so-called reaction. Some people call them rates, but the nature is actually it's a flux. A flux is in general not a spatial flux. In that case, it's called a reaction flux. And how to write this? There are multiple ways to write this, not unique from out-of-term thermodynamics. One way is to write it like this. That's one possibility. Okay? Okay, what is this? The case here is the same K. This is called the reaction rate coefficient. Yeah, it tells me simply how often I turn over, make this chemical step per time unit. Yeah? And then I have this funny e to the mu bar. Yeah? There are different ways to separate these functions symmetrically here and there. It can be all compensated with the fact that the reaction is, that's why I call it coefficient, not constant, depends on composition, okay? This entire physics of nucleation barriers is embedded in this coefficient, okay? So if you have an idea, what is the nucleation landscape of two reacting molecules, you can estimate this from experiments, use Kramer's theory, and run the entire wheel, and then this reaction rate coefficient gets fixed. Can someone tell me, like, why do I write it like this? So what is the constraint I have to obey for chemical reactions to make them relax to thermodynamic equilibrium? So we have now an out-of-equilibrium situation. It's homogeneous, so they just react. So we start at concentration values that are not at equilibrium, and then they approach these concentration values, for example, here, okay? So what is the... Okay, say I had ignored that, and I had just written, this is the forward pathway. Now I go from here to here. You would have written that one? Likely, right? Because you say, oh, okay, I react from here to there. That should depend on the concentration, how much I have here on the left-hand side, and the K forward is just the rate, okay? And here just vice versa. That sounds like the dilute mass action law, right? Well, that is the dilute mass action law, right? But is this K forward and backward rate, are they kind of connected? Is there a constraint? Can I choose any combination of these rate coefficients? Yeah, if you want to do a model, fine. But if you are grounded in thermodynamics, these are real molecules. Can you? Yeah, energy, we don't have link to energy because we didn't discuss temperature transport here. But the fraction is fixed. By what? Yeah, there's the reaction rate coefficient, it's capital K, right? So you're not allowed to choose any combination. But moreover, it's also wrong, because this is only true for dilute systems. And this slave relationship that for dilute systems of the forward, so of K forward to K backward, so for dilute systems, this was dilute. It's just a reminder, so that K forward over K backward is capital K, and this is a constant. This is set by the molecular properties of your molecules. That is an important slave relationship, and only if you obey that, you relax to thermal equilibrium. Otherwise, you relax to something. That is not an equilibrium state. And the equivalent of a non-dilute system is exactly this law. And the relationship I'm talking about is called detailed balance of the rates. It's a relationship between the forward and backward pathway. Yeah, and if you divide through it, you lose the kinetic prefactor K, and you're just left with this exponential mu bar over KBT. For this simple AB process, this is the detailed balance of the rate relationship that you have to obey to make the system relax to equilibrium. How do we see it? Equilibrium is mu bar being 0. So if mu bar is 0, I see that the forward and the backward pathway cancels. Which means there's no time change of the components. And if you don't obey that relationship, you break that. Then you say, oh, I found some equilibrium state where there's a time derivative of the concentration field that is non-zero. Okay, this is what you don't want to have. Good. So that is everything what I want to say about chemical reactions that reaction at react equilibrium. Let's talk about when you maintain them away from equilibrium. Good. So how do we do this now? So as I said, when you maintain a system out of equilibrium, you always have an open system. No matter what you do, no matter what active matter people tell you, they might not model this, but it's always the case. We allow in our case now the exchange of fuel and waste. And we call fuel F and waste W. Okay? It's just our specific way how to do it. Yeah. We want to write both pathways that are possible. One pathway is reaction 1, which is simply our original pathway. Okay. The corresponding equilibrium, even though we don't wanna reach it, is as we learned, mu bar being 0. Okay. The second reaction pathway, so this can happen spontaneously because thermodynamics tells you it's possible, right? Is exactly the exciting pathway where A can meet the fuel and turns over to the molecule B and releases a waste. The corresponding equilibrium, well, sorry, the corresponding equilibrium, well, should be related to the Kemp potentials this plus this minus this and this, okay? And this I here quickly write as mu bar plus delta being 0. And the delta I define here as the Kemp potential difference between the fuel and the waste, okay? That's a modified equilibrium condition. Huh? So if I would not open the system, this is just another kind of chemical system that would relax to thermal equilibrium. But to maintain it out of equilibrium, one way is to fix delta. Okay? Which means there are few molecules inside. They get turned over, and you suck away the waste and always provide new few molecules. There's a continuous flux through the boundaries. Thus, there are also continuous fluxes in general inside the system. Yeah? That's exactly a model for us, right? I mean, you can see like the buffet. There's the fuel. You consume the fuel. It's kind of maintained. Someone always brings the fuel, the food, right? You always consume. And okay, yeah, you leave some waste there as well. Good. We can conceptualize this now with a more complicated model. Yeah. In principle, it has 4 components. So CA, CB, CF, and C waste. Okay. You don't want to do this. I mean, in our papers, you can see this. You can literally integrate out these degrees of freedom if you want. I give you a simple argument that is completely self-consistent. Remember when we had a ternary model, we had all these couplings between the components with these chi parameters. Remember that with the ternary model? Now you have just 4 components, so you get chi parameters among all type of interactions in general. Yeah? However, we can approximately decompose this into a free energy where C is equal to Ca. So it's just our old model, our simple binary model that we had already, and separate these free energy contributions for the fuel and the waste. And this is, of course, in general not possible when the fuel and the waste has some interactions with the other components. So one way to get rid of those is to assume that the fuel and the waste is dilute. Yeah, this is just to illustrate the concepts. You could say like, yeah, but this is in general not true. I agree, but solve the full problem. We did this as well. I can give you a paper on that. But you can still create experimental conditions to make this true, because you just add not too much fuel and make sure that the chemical reaction inside is fast enough to keep the fuel levels inside low. And also when it produces waste, that you remove the waste fast enough that the waste levels are low. So you can actually experimentally also realize these conditions. So it's not an academic limit, but of course in the lecture it allows me to illustrate the concepts more easily. Good. This means we can reduce the problem to a simple transport equation. A transport equation we have already seen with a mobility lambda and the gradient of the chemical exchange chemical potential. Plus, yeah, but now the equation is slightly different. So that's the equation you, up to this point, maybe I add here another bracket. Up to this point, you've seen this. This is the Cantilliard equation for a binary system, okay? And I can do this because I diluted away all the extra components here, and the CA and the CB are related simply by, maybe for clarity, I can write nu A times CA. This is the volume fraction, is 1 approximately. If the others are dilute, nu B CB. Okay, this we had already. Okay, and of course, in general, you would here subtract then the volume fraction of the fuel and the waste, but they're dilute, so I can neglect it. So the new bit is this, right? There's no more Cantilliard equation, called Kahn-Ono equation. But before we get into the names of the history, there are so many names to mention. But essentially, it's a Cantilliard equation with a sink and a source term. We can now decompose S here into a forward and a backward pathway. And it can also decompose each of these pathways into the one associated with reaction, I apologize, yips, 1 and 2. Yeah. So I have here 2 forward pathways. And how do I denote the reaction? The reaction I denote here. This is really, it's not so easy to see because like actually my chalk doesn't work so well on that board. Maybe I take also a new one. This is the reaction type. We have reaction 1 and 2. Yeah. Okay. So I just really make sure that I have all the events of all the forward pathway events to create essentially B molecules, through this pathway 1 and pathway 2. This is this and that. And this is just the opposite pathway. Okay? Good. In general, I can write for each pathway I did a balance the rates relationship. Why is that important? Because if I no more maintain the system out of equilibrium, suddenly you don't do it, you stop eating, you have to relax back to equilibrium, which would mean you die in that sense. Yeah? So you don't maintain our equilibrium, so it needs to be self-consistent with thermodynamics. By the way, in case you have seen like why I have not seen this earlier in context of reaction diffusion systems in pattern formation, simply because people in general do not write down thermodynamic consistent reaction diffusion models. There is a reason, and also valid reason not to do this, because systems are always super maintained away from equilibrium, and they can never go back to equilibrium. Yeah? These systems, however, these are chemical reactions in water where experimentally, by design, you have to think about how to maintain the system out of equilibrium. So the experimentalist could just say, oh, I don't stop adding the fuel, or the fuel gets depleted, I forgot to add it, yeah, because you have to pipette it in. So that's why we have to keep it in the model, also in our theoretical description, to stress or to make clear how we maintain the system out of equilibrium. And the beta balance of the rates relationship here has just this extra delta. Yeah, everything is fine. You see, when the delta is 0, things will just relax back to some equilibrium. Good. Yeah, but now, does it still fit? Where do I write it? Maybe right here on the left-hand side. We can now write down the effective theta balance of the effective relationship for theta balance of the rates of the total forward and backward pathway. That's just mathematics now. Just bring it together. So we just write this fraction, okay? This is what I mean, okay? So this I defined here, right? Have a look. Okay, we wrote now this and that, this relationship, that we can plug it in here to build here this over that fraction, okay? And then we get a very beautiful relationship. Not so complicated, so it's 1 plus this is what we get now. That's a very nice relationship. It just tells me that this net forward pathway is unequal to the net backward pathway, yeah, always when delta is non-zero. And delta non-zero means that's exactly maintaining the system out of equilibrium. However, if it's 0, you see, like, it gets a 1 here. 1 minus 1 is a 0. This is all gone here. I'm just left with this classical theta balance of the rates relationship, and I have relaxation back to equilibrium, as we calculated on the board. And this, in this community, is called, because we do it through the chemical reaction, it's called chemically active. Yeah, to just make a bit of a subtle, maybe also unnecessary difference to the classic active matter community, that there's often some momentum that is transferred to on a molecular scale that makes particle propelled, for example. While here everything is isotropic, there is no propulsion, but it's done purely chemically. To be fair, when you look why a filament is propelled in the motility assay, or why bacteria swim, it's also because there's chemical fuel that gets turned over. However, it's a degree of freedom that's typically not written explicitly in classical active matter models. Yeah? And that's fair, and I just don't want to bash it, but I want to say it why. Because when you think about the cell, yeah? Why do you not write down models for the cell where you have an explicit field for ATP, for example? For some problems, this is relevant. But in general, it's not, and we should be happy about this because the cell does everything to maintain the ATP level rather constant. Because if ATP gets depleted inside the cell, so many processes are screwed up, and that's very hard to take care of the consequences. Yeah? So there's a so-called, we call this about chemostatic conditions, and then you can absorb these conditions into these theories and never worry again, and then you see all these thermodynamic inconsistent reaction diffusion models, for example, for the cell cytoplasm, etc. And they make sense, but they rely exactly on that concept. Yeah? Good. What can we do with this? we can actually, we wanted to understand why these chemically active systems lead to these non-disperse emulsions. And in order to do this, there are multiple ways we can solve a continuum model on the computer, but we can also use these, what I explained to you, these narrow interface models. So narrow interface model was a model where we got rid of the smallest length scale of the interface, yeah? And we solve in each domain inside, outside the concentration profiles and calculate exactly how droplets grow and shrink. And what we need to get for that system, I mean, what can we maybe already formulate an expectation? So what did we have as a reminder? When we looked at the droplet radius as a function of radius, what did we get? We obtained a curve that looks like this. Yeah? And it had here this, well, that's a bit too big. So it had here this unstable fixed point. We called this RC. And RC was time dependent and moving to the right. It was chasing the growing droplets to make sure that only one droplet survives on long time scales. So, what can we expect? What should we obtain when we do a narrow interface model for this model here? We have the model already complete. We have the Banzel rate relationship. We have here this kind of equation. We're done. We could solve this on the computer, by the way. To obtain these snapshots, you just solve that equation. Yeah, but then you see it, you didn't understand. But our interface models allow you to understand what's going on, but how should this graph look like? Can someone draw it maybe? If you make it up, you make up science now. So, but how does it have to look up in order to explain a non-disperse emulsion? Can someone try maybe? Give you a color. You also get this one. I don't know how to draw with that one, but maybe you. Does someone want to try how it looks like? You can't be really wrong. I mean, okay, but the problem is here. Look, we have an unstable fixed point. What does this must have? It fixes size, right? So we need a stable fixed point, right? No one wants to try? No? Actually, this fixed point is hard to remove, by the way. This comes from surface tension. The curve that we will calculate will look very similar. We'll also have this unstable fixed point, but it will go down like this. Okay? Which means if you have a droplet that sits somewhere here, it will grow, end up here. If you're too big, you also come back. And let's do it for each droplet, and then you see these more dispersed emulsions. Yeah? In fact, it's a bit more complicated when you look at more components. Yeah? When you have a conservation law between A and B, and you account for the solvent. Yeah? But in a nutshell, something related happens. The conservation law just makes it a bit more complicated to calculate. But in this very simple system where it's just A and B, in this simple model that was investigated, for example, in the first reference by David Zwicker, it's actually exactly, this is the underlying mechanism. Yeah. Good. And then let's calculate it. And so what do I delete now? The equation I keep, maybe? Exactly, I only delete first for this board. You have a question? Yeah, please ask.
On the graph, the stable fixed point is for the growth, right? the tendency of the droplets? I'm not sure if I understood.
Exactly. So the line I was drawing first was the classics one that undergoes the system undergoes Augsburg ripening. This gives you the radius growth velocity, yeah? And what we need for droplets to arrest at a certain size, we have to make sure that the time derivative of the radius becomes 0. Yeah, that's exactly the case here. So here it's 0, yeah? But it's stable. Yeah, we had a fixed point here already, but it was an unstable fixed point. So anything to the right was pushed also then away from that one. This is what I meant by chasing. So this critical fixed point was chasing because it was getting bigger, the big drops. However, here, when you have this stable fixed point, droplets that are below, that will grow to that fixed point and then get arrested, and they stop growing. They just hang around there forever. And same, when I just put in the simulation a too big droplet, it would then shrink to this size. This is what it means. Yeah? This is what these arrows mean, that one typically draws here on the x-axis. That this means that this one means that the time derivative of the radius is negative. That's what the arrows mean. And here the time derivative is positive, and you see this because the function here is positive, while here it's negative. This is what it means. Other questions? Good. Then let's do this narrow interface model. So what we do in these narrow interface models,
Sorry,
we have
there is a question here.
There's a question? Oh, yeah, please. Yeah.
Sorry. This stable fixed point is at non-equilibrium, right?
Yes.
So do the droplets still maintain like the equal size even when you're actively putting energy into the system?
Yeah, the prerequisite for the emergence of the fixed point is that you exactly maintain it out of equilibrium. If you stop doing this, this fixed point is gone, and this just doesn't exist. It goes to infinity in the end. This one can show. But I think we derived that equation. We now calculate exactly, what did I call it, R star. We calculate it now. That's the point. Yeah, and then you can see it.
This is a way to control the maximum.
Yeah, exactly.
This is a way to control the maximum size of the droplet, right?
Absolutely.
Okay.
Yeah. So when you then measure in your simulations from this continuum model, for example, the size is there's sometimes a geometrical prefactor from the calculation because we have to do some approximation. In particular, the calculation I showed today is the simplest version ever. It has some slightly wrong prefactor. But ideally, when you do a very accurate calculation, also a longer one, this size is exactly the radius of these red droplets here. Good. So, narrow interface model, the idea is we get rid of the interface. We supplement the interface with a boundary condition. Up to now, you have only seen cases, and we also continue this for today, that we assume that there's local equilibrium at the interface. Local equilibrium means that the Kemp potentials are equal, and thus we know their concentrations. And in the respective bulk phases, we simply do a linearization. So what happens if we linearize this? I mean, this we have already seen. When we linearize this part, we just got a diffusion equation. So there is nothing different here. So if you linearize, you get here what I call a diffusion coefficient, Laplace C. Okay. What do we get here? I mean, this is just a local term. Okay. We get the linearized version of that one. Okay. I can write here whatever S ln, for example, of C. Okay. So this we can write it first maybe and then say what the equation is. S ln of C. And what do we get after the linearization? So we have two domains. When we linearize, we get a constant offset, gamma in, and a linear term. And I linearize, of course, around the respective equilibrium concentrations of inside. This is inside. And I can do this also outside. And I can also define these coefficients. I use them as nice shortcuts, not to have a too bulky notation. This is, of course, nothing than the sink and source term S evaluated at the respective concentration I expand around. And the K's are simply the slopes evaluated at these concentrations, okay? But I don't want to always write this with these evaluated at. Good. So this equation has a name, famous in the literature. I already said it today in the lecture. You can just shout. What is that equation? Here we have some reaction terms, constants, and stuff. I've seen this in many books already. It is a. Yeah, just say it out. Exactly, it's a reaction diffusion equation. Yeah. Okay, no surprise. I mean, this was just a complicated reaction diffusion equation, if you like, but we had these. No one calls that reaction diffusion equation because it has these fourth order derivative in the gradients and these, arbitrary, complicated, thermodynamic grounded chemical reactions. Good. A reaction diffusion equation comes always with an important length scale, and this is called the reaction diffusion. And of course, we have many. The reaction diffusion length scale, I write for the two domains. I call them L in, out. The two I can write, if I like, is simply D divided by, cool, because we linearize it, we have reaction rate. This is what we need for a reaction diffusion length scale. Is simply the diffusivity. It's meters squared per second. This is a rate, 1/2. This here is a meter squared. Square root gives a length scale. The reaction diffusion length scale, the interpretation of it is as follows. So when I look at my problem on different length scales, say on the length scale of the droplet size, on the length scale of the distance between two droplets, these are relevant length scales. There's somehow here the reaction diffusion length scale, in, out, respectively. When you're above the reaction diffusion length scale, reactions dominate. And while below, diffusion dominates. And we will use this trick because we will assume now in this simple analytic approach that outside the distances of the droplets is big enough, so we consider such a scenario where the droplets are small. We want to explain exactly that they're organized here in this small dispersed kind of setting. We would like to assume that the reaction diffusion length scale is bigger than the droplet size. So the droplet size is somewhere here. But the interdroplet distance, so this is here droplet size, and I have here an interdroplet distance, that sits somewhere here, okay? This allows us to do a very simple calculation. If you look at this movie, you could say, not so convincing, not so sure whether the reaction diffusion length scale sits exactly in between, completely valid point. That's why the actual calculation is a bit longer than what I present today, and then you can account for these changes as well. And it leads in the end only to prefactor change of this. The coefficient is simply then slightly wrong by the numbers. Yeah, so this is what we do, and this is what also reaction diffusion length scale means in general. Good. Yeah, then we can calculate it. And we get this stationary radius out. The thing is, with these linearized reaction diffusion systems, you can always, in a quasi-static approximation, you can always calculate it. I mean, now you've seen this a few times. I know it gets a bit repetitive. Why I'm pushing this technique so much, because it allows you to understand what's going on. While a canhilliard simulation just gives you nice movies. Yeah? And the general equations, or I just deleted it, are so complicated that it's extremely important to have some analytic approach to it. So let's discuss growth of a single drop, now a little bit shorter because we had it a few times. We can, for example, define here the droplet volume and rewrite our equations simply for the droplet volume. When you do this, you get exactly these flux difference over the concentration inside. I already here neglected again. This I've done already a few times, this concentration outside. Yeah. We now separate the calculation into an inside. Yeah, inside I said it's, well, there are some gradients. That's exactly what I skip now. I don't calculate these gradients. I just say, okay, inside we approximately close to this reference equilibrium concentration. And this is okay, as I said, if the radius is smaller than our reaction diffusion length scale inside, then diffusion dominates, profiles are rather flat. They're not exactly flat, this leads to this correction. Then I can calculate this flux inside, which is then exactly gives me the number of produced molecules, then I can simply integrate, integrate over what do I do here? D3x Vd of my sink source term evaluated inside. Yeah? Maybe I have to write here C. Yeah, exactly, C inside. Yeah, exactly. And this is super easy because if this is constant, yeah, then this gives me just a constant, yeah, but it gives me also a volume. So this is exactly this very simple argument. It gives you just gamma in. Okay, that's easy, huh? And it comes with a very good intuition because you say like, yeah, but how many molecules in total are produced in the entire volume? That is the local density. You integrate over it. That should scale, of course, with the volume. Yeah? The bigger the volume, the more molecules you produce. Okay, so this is one very simple argument. And the other one, we can do the same thing outside. But outside, I go now to the opposite limit. I now assume, as I explained here on the left-hand side, that droplets are far apart than the reaction diffusion length scale. What does this mean? This means we are somewhere here. This means reaction dominate. This means that the far field concentration C infinity is set by the chemical reactions and not by diffusion. So they create exactly this. So what allows me this to do, we can just say, okay, I know how many molecules are produced per time. I can plug in now my far field concentration, C infinity, yeah? And I can simply say, yeah, this is approximately 0. Okay? So which means when you want to calculate that concentration, you just have to balance the chemical reactions and you can kind of neglect the molecules that come through diffusion. Yeah? That's what that means. It's exactly baked exactly into this definition what is a reaction diffusion length scale. And where do we have our S? Have I deleted now the S? No, CS, exactly. Yeah? So this means in the end we just put this to 0 and plug in here C infinity outside. So this one is 0 and we plug in here C infinity. So I can solve for C infinity. Okay? And I get then gamma out over k out plus c out 0. Okay. So this entire thing here allows me to calculate now the flux J out, which I need to plug this in here. The J in I have already, so we're ready. We're almost there. And as we have seen, this is kind of replacing the supersaturation levels. This is as if I replace my supersaturation level epsilon. What was epsilon again? Epsilon, as a reminder, was this excess concentration with respect to the equilibrium concentration, -1. Yeah. And where did the epsilon appear? Yeah, the epsilon appeared in my transport equation. Now I integrated already over the surface. That's why I get a 4 pi dr, yeah, C out 0 epsilon minus L gamma over R. Yeah, so this equation you've already seen. I just multiplied by R squared. That's why I have an R here and a 4 pi. That's the only thing what I did. Just in case someone wonders why does he write here capitalized quantities instead of small ones, this is because I integrated over the surface. Yeah? So these are the integrated quantities. Good. So we're done. We can now write a growth equation. Yeah, an approximate one for sure, but it covers all the physics. Just when you compare it with the simulations, you can work a bit more precisely and get a few prefactors a bit better, but you get the same physical dependencies. That's why I write here really also approximate. So the equation you get looks similar, first of all, structure-wise and prefactor-wise as we had it already. So this prefactor we had already in our growth equation, right? And then we get a supersaturation level that is now set exactly by the reactions. We have the surface tension is hard to beat. It's always there. It will always set the critical point. Sorry, critical radius, not critical point. And then this is really the new contribution that comes from the J in. You see? So what is new compared to the equation you've already seen? This term is new. It comes from the inside. And this term, which is a negative supersaturation level, the entire term, yeah, is also new. And it came from the outside, because far away from the droplet, the chemical reaction now fixes the amount of supersaturation. Yeah? And so it keeps on producing the material that then flows into the droplet. The droplet gets deactivated and goes outside. And so the size can be balanced by this circular flux. Everyone who loves mathematics can now solve this. So this is dtr. Where do we have the r? Here in r, that is not so important. That's a constant. This is 1 over r, this is r squared. It's a cool problem. It has 3 roots. Lots of unphysical ones and more precise complex roots. We don't have to worry about those. We can do a simple-minded argument. So we put it, of course, to 0 because we want to calculate exactly these, as I said, the roots. So these, the unstable and the stable fixed point. Yeah? And there's a very simple standard argument because, we don't want to mess around with standard algebra. I just rewrite it in a more simpler form again. So if I put this to 0, I recast this epsilon, then the bracket. This bracket has to be 0, right? This is exactly this one. I wrote here r. I multiplied by r. So I get here an r epsilon. So this is epsilon here, this entire thing. Okay. Yeah, exactly, it's epsilon. and multiplied by r, so I get just an L gamma here. You see this? And here get an r cubic. And then you could say I can look at now two limits. Mathematics is a famous name how to do this. And large r. So for small r, yeah, small r, this is not relevant. So what has to balance is this with that, okay? To leading order, okay? So to leading order, the first root gets approximately r gamma over epsilon. For large r, I mean, this is a constant, they have to balance each other, okay? And this gives me the second root. And let me just write it. It gives us square root minus 3d our epsilon, which was just a shortcut of all these beautiful parameters. So I write it here explicitly. That depends, of course, on these active parameters, gamma out, k out, and it gets divided by gamma in. Okay, cool. So we found an interesting root. Okay, there's a negative sign. That's not so good. That's fine. D is positive. This is positive. The epsilon we defined here with a minus. This is also positive. I can tell you in a second. But the gamma in, for this to exist, needs to be negative. Physically, what does this correspond to? You have an A. We talk about the direction of A, so that we deactivate A inside. Yeah? Okay? And then the A goes out and gets activated outside. Activated outside means, where's the epsilon? Activated outside means that, okay, let me think. Did I do it right? One second. Should not say it wrong. 1 second. I wrote it somewhere. Okay, out needs to be negative, exactly. And gamma, it needs to be activated outside. Exactly, perfect. Exactly, so it gets deactivated inside. So we have to activate it outside. So gamma out is positive. Okay, so this is positive. This is a positive concentration, but we have to cancel this negative sign. So k out, that's the slope, need to be negative. Makes also sense, because otherwise we have a runaway solution. Look at this linearized term here, outside. So capital gamma out, see like what I wrote here? This is positive, so to produce it outside, let's say the droplet is not there or something else, or doesn't take it up too quickly enough, you have to make sure that the system is balanced. Yeah? Otherwise, the solution just runs away. Yeah? Okay, I'm 2 minutes late, but I'm already done. Yeah, I think I'm done. And now if you plot this solution, yeah, if you plot this solution now, so I hope could convince you that everything below the square root is positive. Yeah? And we have this one remains. This is the unstable fixed point, and we get exactly the yellow curve as we were hoping to get, yeah? With an unstable fixed point that is to leading order almost equal to the critical point, and a new intersection that is set exactly by the balance of the activation outside, the molecules diffuses in, gets deactivated inside, and you have this circular flux. Yeah? So it's a unique result. It's a non-equilibrium steady state that comes from the system being open, yeah, and breaking detailed balance of the rates of the chemical reaction. Yeah? And experimentally, you create this by chemostating, for example, the fuel and the waste molecules at the outside, and then you have an out-of-equilibrium system. And ideally, you see this size control, and if we have had a bit more time and work a bit harder, we would see the shells forming, dividing droplets, oscillating droplets with our second chemical reactions, and we can keep on going. Yeah? But that is the underlying basic idea, is exactly this extra stable fixed point. Are there questions? Please, yeah.
So in your picture on the right, it looks like a hexatic. Does that mean the droplets are repulsive?
Exactly. So the only thing what I calculate is a single droplet analysis. So the single droplet can already control the size itself. Yeah? Now you have to embed this into a collective problem. Yeah? Indeed, it's true. There is some effective repulsion that is exactly created by this coupling through this outside concentration field. Yeah? One can show it's actually a weak coupling, and it leads to a new phenomenon. This is exactly the ordering of the positions of the droplet. Yeah. So this is there's follow-up work actually also from David Zwicker on that. If you want to have, I can show you the paper. But this was a bit too much for the lecture. Yeah, but well spotted. Yeah.
Excuse me, Professor. First off, thank you for the lecture today. And the last explanation of yours, can you repeat why K out needs to be negative?
Yeah.
Please.
Exactly. So, outside we produce the material. Yeah. So when you produce material outside, you need the droplet to suck in the material fast enough. Okay. So in general, this is not always given. So it depends on droplet properties, etc., that it can eat up fast enough. Otherwise, you get lots of unphysical solutions that would lead to a blow up. So if I have not written that term, for example, and put this positive, there might be cases where the droplet is big enough that it sucks in the material fast enough, but if it doesn't, I get an ill-defined solution. But in general, I mean, you can expand to arbitrary order. This exists, and that takes care of all this. So it creates you a much more kind of solutions. And actually, when you construct this, I wrote this out with the balance of the rates. If you actually do this, you find this is actually negative. Very naturally, very self-contained. But you sense a little bit. Otherwise, you lose a lot of solutions that can be physical, but only because you truncate it at series order. But in principle, you're right. This could be also positive, and you go to the second order and then make that negative. That's another alternative. Definitely. But
thank you.
You need a stationary state in concentration outside.
Could you repeat the discussion of this last part? I'm not sure if I got everything. That's
exactly. So when you look at that solution, first of all, it's a square root to have a physical solution. Everything below the square need to be positive, right? But so there's a negative sign here. We have lots of constants that are always positive, like the diffusivity, the concentration here, etc. Right? So we have to carefully now discuss for what choices of either producing inside or outside, this gives you an actually stationary solution. Exactly. And here, when you look at this equation, we haven't talked about the signs at all. I've just told you what the series order term in and out is, right? You just evaluate the sink and source term at this concentration, and depends on your reaction scheme what it is. There are also other cases, by the way. There are also so-called externally maintained droplets where you have a runaway solution. The droplets just grow. I give you a simple example. Say you just have a reaction where everything you activate inside and outside. What happens? I mean, there's no stationary state. You just have super hardcore speed up growth, right? It would just look like this. This is also possible. You just have external matter supply, you just feed the material. So this model can deal with these cases as well. But of course, I was a little bit interested in that phenomenon. So you have to think about whatever you produce in some domain, that you kill it in another domain. So that you create this asymmetry between activation and deactivation, between the inside and the outside. And then now, if you now make a choice of the signs, say you, we said, so the gamma in to be negative, so we deactivate inside. So if this is negative, we have to activate outside to have even a chance to balance this situation, to have a stationary state. And that's then exactly enters this discussion of sign. It's very self-consistent because this one is negative, cancels this sign, and the epsilon, that's why I also defined that this is actually a positive subsaturation level. So everything is positive below the square root. Yeah, yeah. You can have the opposite. So the outside, you activate outside. Yeah? This is, okay, interesting. So, yeah, of course you have, sure. You just swap A and B component, same thing. So no one told you what is A and what is B. Yeah, exactly. No, of course. Yeah, but good point.
The square root has two roots, right?
Sorry, second.
The square root has two roots, right? What happens to the negative root?
Unphysical. It's negative radius, yeah.
It's more comments. It's interesting because yesterday I was talking about this active membrane where you have inclusion, so it's a very different physics, but there are activity as well. And it was a theoretical model, and there was no direct, let's say, so far, maybe a demonstration, but at least the conclusion was the same. So you end up, if you have this active inclusion, so in this case, they don't have a reaction, and also the importance of the coupling to the deformation of the membrane is important. But at the end, you end up with domains of proteins which also have finite size and which are also very regularly distributed in the space. So I think that's interesting to see. For a different type of reaction, eventually you end up in the same type of distribution. I think that's interesting.
No, I think, so from a very zoomed out perspective, I mean, but connecting to the question you just asked, I mean, when you see such a state of these multiple droplets, all of equal size, this can be a pure collective phenomenon, which means that the single droplet would never do this, but as an emulsion, they do it together. In that case, just a very lucky, cool system, but the droplet, each droplet itself is doing it already. That's why also from the analysis, it's so simple to show. And I think the entire, from a very zoomed out perspective, the entire idea of having these ordered lattices of dots, I mean, this is also, it's a common state also in pattern formation. It's rather generic, and that's why when I show you a pattern, and in case you see also some science talk in the future, and someone shows you a pattern and makes a big thing about it, patterns are not generic, right? I mean, this is, do we want to explain a pattern for a specific system, and you want to show that a specific system leads to that pattern? That's the hard job in physics. While from a very zoomed out perspective, comparing patterns between theory and experiments is not meaningful because it doesn't tell you anything. You have to make sure that when you perturb it, and also when you collect all the fundamental mesoscopic or microscopic ingredients that, and symmetries and conservation of that, this is consistent with the experiment. And that's really the hard bit in general. Yeah? So never compare just your figure 5, cool, I have a stripe pattern, and oh, the experiment sees also stripe pattern, so my model is right. Very unlikely, because I can write you, okay, I can write you dozens of models that give stripe patterns, and you have to, but you have to find out as a physicist which one is the right one, which model. That's our job. Yeah. Thanks for inviting me to make this concluding statement at the end. Good.
Well, I think that sounded like a very good closing remark, and also for just to keep things more or less on time, I would suggest we can wrap it up here. Thank you, Chris, again for the talk, for the lecture, sorry.